So what we're looking at right here are 4 chromosomes. So there's 2 pairs. One pair is yellow, the other pair is red, and they're tagged for a reason that will become clear in just a minute. So what we're trying to do is go through meiosis. And this is showing what it would be like in an interphase in a cell that is going to make either eggs or sperm cells in humans. And these 4 chromosomes are in 2 pairs. What would happen, and it's similar to what would happen in mitosis, is that the first thing that would go on is we would have S phase in interphase when we're getting ready to do this. So we'll go ahead and I'm going to pretend that I'm completely going in here and duplicating my chromosomes perfectly. So we're just making exact copies, okay? And so that's what you can see here. All right. Then what's going to happen that's a little different is that when we get to prophase, where it's prophase I and we can see chromosomes, at that moment we will realize that our chromosomes, instead of staying in these pairs with identical sister chromatids, they've actually come together with their homologs. And this is what it would look like. Okay. And what's going on with this is that we've got the red chromosomes duplicated. So there's 2 with orange and there's 2 with blue. But what's really happened is that they're duplicated and now they're sticking together in the middle, making a tetrad. Sometimes it's called a bivalent. And what that means is we've made duplicated copies so we have enough for new cells, but we don't want to put everything in the new cell, because if you have an egg or a sperm cell, you need the other one to come in to make the new person. So that's what's going on. So the first thing that we'll see in prophase I of meiosis is that we make these, and we'll have to go back through to see the other thing that it does separately in a few minutes. So this is prophase I, the nucleus is gone, and I see these tetrads. Then I go into metaphase I, and I line them up on the metaphase plate. And what you'll notice is that right now I've got pink and blue this way and orange and blue, but it could have just as easily lined up like this. And the fact that it could go in either direction is part of why we have variety, because we actually have 23 pairs, not just 2. So we're in metaphase, and then from there, if you've gotten metaphase I set up and you did prophase I properly, it's actually really simple, because now all I have to do is divide what I actually have. So I go to anaphase I and I pull apart anaphase I, and what you're noticing is that there's 2 pieces now, not just one like in mitosis. That tells us we're not done yet. So what we're going to do is end up with these 2 different cells here. Okay, then we go into prophase II, where we can see this again, and then we're lining up in the metaphase plate with 2 of them now. So we've got really this cell and this cell. Then we do metaphase to anaphase. So this is a cell, this is a different cell. I do the same thing over here. Same thing, I've got a cell and another cell. And what you can see pretty quickly is that when we're done with meiosis, I've got the equivalent of 4 separate cells. I've got red and yellow in each one, and I've got 2 different versions. In this one I've got pink and blue, dark blue, pink and dark blue versus orange and light blue, orange and light blue. So these would be 4 separate sperm cells, or one of them might be the egg if it was human. And that's how we get started doing meiosis. Okay, the second thing that we then do is go ahead, and if we did this one more time, we're going to start from the beginning again and do this one more time. And what I'm going to do is have one of each like we had before. Okay, and now you're going to see why we color coded. This is the time that that becomes more clear. Okay, so if this was in interphase, and I just have these chromosomes hanging around, okay? Then I go into S phase, and I go ahead and I make my duplicated copies, just like we were doing last time, so nothing's changed yet, okay? What's happened is that we've got these set up where they're duplicated, but I've got like these yellow ones in prophase I of meiosis are going to hang out like this, and similar idea here. The one other thing that can happen in prophase I besides making these tetrads is we can get crossing over. And if you assume, say, these blue two were copies of the original one from your dad and these were the copies from your mom, if we do crossing over and we swap some of this, we are going to suddenly realize that we've got more variety than we had a few minutes ago. And this is how it would come out. So now I've got a copy that was just like what came from my dad. Now I've got a copy that's part my dad, part my mom. I've got another copy that's part my mom and dad, but different parts. And then I've got this copy here that was just like my mom gave me. That means that all of a sudden I've got 4 different versions going on. And I'm only going to show this in one of the 2, but in reality this happens all over the place. So that was prophase I. We made bivalent tetrads. Then we did crossing over with one of them. We could have done it with both if we wanted. We line up on the metaphase plate in metaphase I. We divide what we have. We're not done yet. We have to do it again. Okay, and now what I want you to see is that now I've got one cell right here that has the one from my mom and another one with orange on it from one of my parents completely intact. I've got this one where I've got the orange on the red one, but look, I've got a mixture right here. This isn't the same. These are different directions for that offspring. Same thing over here. I've got this yellow one and this with some of my mom and dad's on there, but a different batch, and now with the dark blue. And then the last one, similarly, it's different. So what's happened is I've actually gotten 4 different gametes with one set of chromosomes, but each one of them is actually a little bit different. So it's not just which one went where, it's more than that. Okay, so that gives you even more variety. The last thing I want to show you is I want to go back to what happens if we go back to when we had our prophase I, and we maybe did our crossing over. That's okay. All right, I want to show you one other thing that can happen. All right. What was supposed to happen right here is we're supposed to separate. Okay, so we got 2 cells with duplicated copies. We know they're not identical exactly, but they're close. And this would be the cell, and then I would be going ahead and I would be separating these again. And what's supposed to happen, if I just go to this side, it was supposed to divide up like this. Sometimes what happens is one doesn't split. So what if I went like this? Okay, we'll skip this other part. What's going to happen is I'm going to get a cell that is going to have these guys together, and I'm going to get another cell, which is this part. And what that means is that in this one, if it made into a person, and this happened to be chromosome 21, I'm bringing 2 copies. And in this one, I'm bringing no copies. Probably this isn't going to work. And if that egg gets fertilized with the sperm and one of them had this situation, you probably don't get a person. You'll never see that. This one we know we get if this is chromosome 21 because we get Down syndrome. That's trisomy 21. So that's what we're looking at. Is it's just a mistake when you're pulling them apart. And if you pull them apart separately in a way that you're not supposed to, you won't get what you expected, and that can cause some trouble. So that's how we can get differences within just the meiosis part, is having extra chromosomes or missing a chromosome occur, and that can cause a lot of difficulty in the numbers of chromosomes. So you would have had a monosomy where you just get the one red one from the other parent, or you'd have a trisomy where you get one from the other parent, but you already have two. Okay? So that gives us a view of meiosis. The big changes are in prophase I. If you go ahead in prophase I and you do tetrads and then you do crossing over, you got it. The rest of it's just like mitosis, where you split until you're done. And then what was supposed to happen was this, where we would get the 2 and 2. That was what was supposed to happen. And then that's it. And then you just have those cells, the egg or sperm, and they find ideally their associated partner, and everybody gets the full set of chromosomes. So I hope you've enjoyed this little demo of using the beads to kind of show you how the chromosomes move around. And you could do something just like this at home using some yarn or maybe socks. You could do different colored socks. It would all work out. Anyways, I hope you've enjoyed, and I hope this is helpful.